Hello, so welcome everyone to our first meeting for this course, Bioinformatics 6310 or 6310, Introduction to Computational Methods in Bioinformatics at Northeastern University. I'm very excited to be with you today and go through over the first lecture and see the plan for look into some policies, the plan for the whole course, and then start diving into some molecular biology and genetics. So today, some of the things that I'm going to look into in some details is the administrative craft of this course. So things that are of concern to you, such as the grades, the marks, the policies, using some tools like generative models, ChatGPT in particular, and then we'll move into one very important discussion on chemical structures of genetic material, with some highlight of history timeline of showing the development on this topic of genetic material. Then we'll discuss the central dogma of biology, which is very important to capture and digest the concepts of transcription and translation. Some of you might have heard about this. If not, we'll go over it. And then give a context of the different species and look into the types of cells and some of the differences between prokaryotes and eukaryotes. So throughout this lecture and meeting, try to again emphasize some of the important details in terms of the administration of this course, including the learning management system that we're dealing with, the grades and so on and so forth. And then along the way, we'll start talking about some biology, bioinformatics. We want to put some, understand some fundamental concepts that will help us throughout the course as we progress and start diving into more and more about bioinformatics, in particular, the different data that we can handle and so on and so forth. So in terms of this lecture, understanding the central dogma, the nature of this genetic material that we are looking into, DNA, and in fact, the history of DNA discovery is very foundational to molecular biology and modern biology. This is because it provides insights to molecular bases of life, our evolutionary heritage, and also highlights breakthroughs that have shaped the current world we are in terms of biotechnology, technological and medical advancement. So starting with the course plan, two things that I want to bring your attention to on the slides here. Pardon me, let me put the pointer here and go back. Is now we're going to talk about, oops, maybe I would switch to another color. Okay, let's use maybe black for this. So Canvas. Canvas is something that we need to keep an eye on. It is the LMS, the learning management system used for this course. I've personally tried several learning management systems, and I guess the way this Canvas is set up for this course is really attractive and very useful and easy to maneuver through and find all the details needed. And also some important information regarding the syllabus for the course. I'm going to introduce myself and hopefully I get the chance to meet you on the office hours. I'm looking to meet all of you during the office hours to get some information about your background. So make sure you come to the office hours. It is our chance to meet on, one-to-one basis or even discuss as a group My name is Othman Soufan, or Othman Soufan. That is fine if you wanna call me by that. I'm I'm a part-time lecturer at Northeastern University. Um, I'm an assistant professor in St. Francis Xavier University in Nova Scotia in Canada. My background is in computer science, so I hold master's and PhD in computer science with focus on bioinformatics. After finishing my PhD in King Abdullah University, KAUST in Saudi Arabia, I moved to Canada, where I've spent roughly more than 3 years in McGill University working on bioinformatics research in SIA Bioinformatics Lab. It was a very exciting journey where we had collaborations with a big project where we had collaborators from Environmental Protection Agency in the States, Shell, the well-known oil company, Environment Canada, University of Saskatoon, and so on and so forth. I've published many research papers and still excited to work on more problems. I have 15 years of experience in data mining and machine learning, but roughly 10 years of experience in applications of AI and bioinformatics. So hopefully, again, we meet during the office hour. And again, I would really like to invite you all to check the syllabus and see the timing for the office hour. I'm going to set the schedule also for the office hour on Teams. So keep an eye on Teams as well, the post that we have on Teams. Coming into the office hour, give us the chance to discuss and also know something about your background. So let's go over some principles for this course. You know, what I'm looking for is, as you join this as graduate students, is to have a journey in this class. Okay, I don't want you to come here and start getting worried. Oh, am I going to get the marks or not? Am I going to am I going to lose marks? This is an easy or hard course in terms of exams and assignments. I want the way that you come to this course is really to have a journey and an experience that you want to go through. I'll do my best to give you exposure to important, concepts, and I would love to share my experience and try to help you to get more excited and see the importance of this topic. My aim would be to have you fall in love with bioinformatics. So I want you to stay excited and look for the coming material and lectures and so on and so forth. And I want you to engage with me, okay? So the first thing is, I want you to be a knowledge seeker, okay? So knowledge. Seek knowledge in this course and be curious, okay? Engage, okay? I want you to engage in asking questions through, the Teams, the chats, emails. I want to have communication with you. I want to make sure that, we are at the same point of understanding what we have in this course, what we are doing right now, what's going to happen next, and so on and so forth. So please ask questions. And when it comes to this course, as this is a graduate master's degree, building your skills is very important, okay? So skills, your skills, things that I want to boost your own resume, okay? I'll be proud of you once you have this course and get some project out of it. Have it in your GitHub repository, have it listed on your own resume. This would make me happy because this is gonna make your resume stand out more in the future. So in terms of the course, this is a snapshot of the option panel in Canvas, and you would find there's a home page. There is as well the syllabus as you can see. Oops, sure, sorry. Okay, so there's the syllabus here. There's list of modules. I'm going to talk about that. So it's going to show you week by week the material that you have access to, and all related tasks for each single week, people grades, Zoom meetings. So keep an eye on Canvas, adopt it, bookmark this Canvas page. Try to visit that every now and then. Keep an eye on it, okay? So it is very helpful to organize the course material. It is the main way of sharing the content I want, and it serves very good communication between you and I. So if you click on the syllabus page, what you are going to see is the list of material and topics that will be covered throughout the weeks, okay? So for the first few weeks, you can see this is going to be more biology / genomics oriented, and then we'll start moving to some more computationally condensed parts, including high performance computing and sound processing and of RNA-seq data. Then there will be a break, the midterm break. After that, we'll talk about some, to highlight interesting applications for all of this material. It makes sense to bring attention to topics like cancer genomics. There will be a special topics where a guest speaker is hosted. And towards finally, the final presentations, we'll be working as teams. For the grade, which is something that I guess many of you would be wondering about, how is this course going to be graded? So here you can see the marks distribution. For assignments, you'll have about 40% quizzes, 25%, discussions 15%, and the final group project is about 20%. This distribution of marks makes it difficult for you to skip any. Meaning, if you skip any of those, you are going to certainly miss the highest grade in the mark in the course, which is letter A. Okay, everyone is hoping to get to grade A. The focus here is on trying your best, and then again, try to meet the deadlines. My advice for you would be, make sure you watch the lectures, come to the office hours, ask the questions that you need to ask, and meet the deadlines by starting early for studying for the quizzes, doing the discussions and tasks assigned, and also planning for the assignments. Okay, final group project. This is going to involve some paper selection that you'll be working on reproducing the results for, which is something I believe is cool to have as a project. We are aware of challenges in doing that, but again, it's going to be a very interesting and nice experience for you to learn about. We'll be talking about more about the final group project in the coming weeks. This will involve several steps, including paper selection, individual write-up, group presentation, and peer evaluation. Peer evaluation is this dedicated percentage where your classmates are going to evaluate your presentations and the deliverable, the results you deliver in these presentations. So I would still highly recommend and ask you to go and download a full version of the syllabus. If you go to the home page in Canvas, and then you have here, you can click on the syllabus, and then you can download it from here. Okay, you can have a full PDF version, so store that somewhere. Read the syllabus. There are very important information that are not all covered here today. Okay, so I highly recommend and would like to advise you to go and download the syllabus and fully read that syllabus. And if you have any questions about the syllabus, please feel free to reach out to me. Modules, two modules that you can start with. or module S that would help you to set up the needed software if your operating system is Mac or Windows. Check those. If you have Linux operating system and you're struggling to figure it out, also try to reach out to me as soon as possible. And there is module D. This is discovery orientation. That is, discovery is the name of the computing cluster that is offered for us to use in Northeastern University. So it is a very good resource for the students of the course. And it's really nice addition to this course that you will have a resource, available resource to run heavy computations or to, host your data and run the different programs. I've requested for an assignment of discovery account for the course. I'll be talking about this also later on in the coming lectures. And one thing to remind you about is this going to go step by step. So you go through module S and then module D, module S and then module D, and that is the workflow. And then you'll have module 1. So before jumping to module 2, for example, which is going to be week 2, you're not going to be to go to week 2 until you click on week 1 and skim through and read through the the information there. That is very important for us to make sure that you are giving yourself the chance to check all the important details that we want you to receive. Another thing is to keep an eye on what? The due dates, guys. You see here the due dates for all coursework. In this course, basically Wednesdays are going to be the deadlines. Okay, Wednesdays are going to be the deadlines for delivering assignments and quizzes in general. The due dates, you can see the due dates on the modules tab. Okay, so when you click on modules tab, you can view the due dates. There's the to-do here widget that you can see on the home page. Is also going to give you some marking and highlight of the due dates, and if you scroll down to the bottom of page, also you would see the due dates. So there are different places where you can access the due dates. That is why there's no excuse for anyone to skip any of those. So it's very important. And from now, I'm telling you, all the due dates are in general going to be Wednesdays at 12 p.m. EST time zone. Okay, Wednesdays. Mark it down and know it. So assignments, quizzes, discussions, due dates would be Wednesdays. People tab, okay, or people link on the Canvas home page or Canvas. There you will see the group that you are assigned to, because as we said, the final project is going to be a group project. Okay, you'll be working in groups. And here you would know which group you are assigned to. It will be helpful for you to have discussions with your teammates. Now you can use the groups feature in Canvas to do the discussion, or you can use Microsoft Teams or any other means or medium to communicate with your teammates. Now, in terms of asking for some help, when the time comes and let's say that you are facing some challenges, in general, I welcome communication. So I hope we will have an active chat on Teams, okay? And when it's needed, please email me. I've put some instructions in the syllabus regarding the email. Now, if you have some technical issues, some coding challenge, okay, so and you need some support, these are the steps to follow. So I'll quickly go through them, but I highly advise you to read them when you will see the lecture and access the slides. You have to send the code. It's not about sending screenshots, okay? You have to make it easy for me or the TA to go and reproduce the issue that you are going through. And give some description. What is the input and output? What is the error? And this is going to come with an assumption that you have already tried, right? So it's not like the first moment you have an error, you're going to email this to us. You have to go and try. You have to develop the skill of debugging and looking on online resources, okay? And so on and so forth. However, if that may not be the case and you have not been able to solve it on your own, feel free to share it with us, maybe through communication or as a GitHub issue tracker and so on and so forth. Another thing that we decided in this course and as part of the discussion with program management is that we are going to embrace generative models. And personally, I teach other courses in other universities. I also ask students and let them know that they are allowed to use generative models. On top of that, it's very common nowadays, ChatGPT, okay? ChatGPT, and there are different versions, GPT-3, GPT-4, with difference in terms of performance. So now, these are certain instructions that we want you to follow if you decide to use the generative models. First, we want you to know that you're not required to use generative AI. So if you're not using it, you're not missing anything necessarily. If you use those, you have to cite, you have to mention that you have used it. And the way you cite that is to refer to it and then give more information like which generative AI was used, what prompt you have used to generate the response you get, why are you using it, okay? We are hoping that you have a reason why you're going to use ChatGPT, okay? It could be, that you want to find an answer for a specific question that you couldn't find easily, right? It could be that you are trying to generate a line of code because the library that you have not used before, okay? The idea is ChatGPT is not here to replace the learning efforts that you need to put in this course, okay? So a justification is needed. And by the end of the day, we are going to all have, approach this with good intention and hope that everyone is having the goodwill. Do not misuse ChatGPT. Do not hide it from us, okay? This may result in retracting the use for the generative models from the whole course. And if this happens, it would then mean that when you are using it and you're not allowed to, that this is an act of cheating, which I'm going to talk about. And this is one thing I take personally very seriously, and that the whole department takes and the program takes it very seriously. In terms of late work, my recommendation is to meet the deadlines. Do not miss the deadlines, okay? Unless you have really, a legit excuse of why you missed that, and hopefully that's going to happen before you miss the deadline. I believe it's going to be difficult to accept delays without penalty. So the penalty is listed here in the syllabus. Again, I highly advise everyone to meet the deadlines. What about pre-submission? If you meet the deadline with your assignments and submit on time, you have the chance to go and improve your grade within 1 week of the original grade, okay? So this gives you a chance, if you have time and you're really interested, to go and refine your work to solve a specific problem. And so that's welcome. That is welcome, okay? Because it's another chance for you to learn, but to the extent that's not gonna affect your other submissions. So make sure you understand that and know that this is not gonna be a full mark, graded out of full mark. It's gonna be out of 80% of reclaimed points. Now, cheating is not accepted, okay? And I'm telling this to you very clearly. It's zero tolerance, and I hope you as graduate students, you are already mature enough to know this. We may use tools to see if you have used, copy pasted this from each other. If there has been an act of plagiarism, I personally, when you are going to submit any piece of writing, I may very likely go and run some tools for plagiarism check. And if you have been found to copy and paste, this is going to be reported directly, okay? So you can read more about this in the syllabus, but this will be reported to OSCCR office, and this may result in you receiving 0 for the whole assignment, or maybe an F for the whole course. I mean, the whole process will start to decline once a student is going to cheat. We should be all committed as you come to this course, right? I am here to teach and learn at the same time. I'm not just here to give one-way communication. I'm here also to learn from you. And the way that you are going to teach me is through, delivering the solutions to your assignments, doing your discussions and writings, working on your projects. The atmosphere here is where we want you to build the proper skills so that you'll be ready to the market, okay? And go and deliver on real life experiences and real life and show impact on real life projects. So if you establish a way where it's going to be, I have to sneak and find solutions, cheat from here or there, copy and paste, is not healthy. It's certainly not healthy. So it's not personal, but we're not gonna tolerate this, and we take it very, very seriously. So please understand this. May you face any discrimination, there are clear policies in Northeastern University, so you can reach to what is called Title IX. And also there's another thing I want to highlight is Family Educational Right and Privacy Act, which means that your own grades and marks are going to be kept private for the course, okay? And more details on this in the syllabus. So that's it for the administrative craft for the course. To summarize here, I want really to emphasize a few things. That is Canvas, okay? Canvas is where you'll find all the course material. Microsoft Teams will be, I've already created a team for us there. So it's going to be, a good place for posting the recordings, and also on Canvas, mainly for chatting, okay? There are conversations that we can have, so please use that to send me messages anytime. I respond to these messages once I'm available. Another thing is academic integrity is very important. Academic integrity in this course, please do your best to take this as serious as it could be. No cheating is to be tolerated. There are other policies regarding the assignments and using generative models and ChatGPT and so on and so forth, and grading of the course. You should now have a clear idea. If you have any question, if it happens that you have any questions about the course material and syllabus, please reach out to me as soon as possible. Okay. So, chemical structure of genetic material. And with this, we want to talk about the timeline in history. And there are certainly some important dates showing the progress of research on releasing what is the language that is in our human body and other types of species, including animals, plants, and so on and so forth. What is that molecule of life? What is that genetic material that we have? And what information it includes is key to understanding diseases, underlying mechanisms of diseases, key to develop treatments and medicines. So knowing the history of how this was developed in terms of, research and discovery of the genetic material should be really appreciated, because this is helping the humanity now in all aspects. So the first thing that we want to go to is to give the credits to very important names here. Okay, the first name here is Miescher or Miescher. Okay, this scientist. Miescher is a forgotten name. Okay. Just give me a moment here. Just missed his first name. I think it's Jonathan Friedrich Miescher. Okay, so Friedrich Miescher is a very important name in the history of discovery of DNA. That is generally is, I don't want to say ignored, but not highlighted enough. So Friedrich Miescher has a very interesting story. Apparently, he studied medicine. He got typhoid infection, which affected his hearing, and then he couldn't practice. He couldn't become a physician. So he decided to go to research. And part of his research, he wanted to dive into the molecular or chemical composition of what's inside the cell, the nucleus. So he asked for bandages for the pus. From I think from one of the pharmacies or hospitals, he had asked them to give him these bandages with the pus coming from the wounds or some of the health conditions. He would take those and start by applying alcohol wash, which is going to remove the outer membrane. And then this is the beauty, where he spent lots of time trying to explore and try to get the material inside the nucleus. Okay, so he used pepsin wash. So he took this from the guts of the pig, and then that's helped in removing the cytoplasm, and then used base wash using baking soda, which ruptured the nuclear membrane, releasing all the material inside. Okay, and then finally made some acid wash, where he ended up with the white precipitate that was formed in the tube. Okay, He didn't know exactly what is that by then, like what is the composition of that white precipitate. Uh, Apparently, it was the impure version of DNA, and he referred to it as nuclein. Later on, Altman, a student of Friedrich Miescher, Altman uh, looked into, revisited some of the work, and in 1889, he referred to the nuclein that Friedrich Miescher discovered in earlier. That is around 18 when he published his research, 1871. So Altman came up with the naming of nucleic acid. So he's credited for that research and pointing the acid term for the DNA. It's not yet the DNA at that stage, but actually that was the impure version of DNA. Later on, Albrecht Kossel looked into the chemical composition of nucleic acid. So now there's this material which is at the core of the cell, and Albrecht Kossel did the research to try to find out what is inside that material. What is the chemical composition in particular? This is when he explored adenine and termed those adenine, thymine, and cytosine. Now, guanine was already isolated by another scientist called Julius Bodo Unger in 1844. And so that is not necessarily credited to Kossel, although it was characterized, okay? Now, an interesting piece of information that adenine, thymine, cytosine, they link to their sources, okay? So adenine is the Greek word for adenos, which is the pancreas. This is where it was isolated from first. Thymine was isolated from the calf's thymus. That's why it's called thymine. And cytosine is derived from a German word called cytosin. And this is part of the Greek prefix of a cell, which is called cyto. And that's what we refer sometimes into, in cyto screening and so on and so forth. So more advancement at that stage in terms of research and looking into what is this genetic material. So there was this continuous effort to try to, reveal what is that material that we have. Later on, we have also a notable breakthrough, and that would come later on, okay? So Asbury and Bell, these two scientists you see here on the left side, they were interested in looking into the X-ray crystallography and trying to characterize the DNA. They want to try to unravel and reveal the structure of the DNA. X-ray crystallography is still a very important technique and technology to dive into the compositional structure of molecular material. Also, this was followed by research and by Wilkins and Franklin. And the work they have done was very prominent and needed for the discovery of DNA double helix. This photo that you see here was labeled photo 51, and this was taken at certain stage of the DNA So there are some more details that you can read on terms of the history or the reading material provided for this week. However, these photos were really important into bringing the thinking, okay, now we have this nucleic acid, what is the structure of that material? Later on, a major achievement credited to James Watson and Crick, okay, Francis Crick. And again, preceded by that was these efforts on X-ray crystallography, which actually helped in the determination of the structure of the DNA, because the photographs that Rosalind Franklin, the lady here, that produced the photographs that helped and provided evidence that helped James Watson and Francis Crick to propose the following key terms: the double helix model of DNA. Okay, so that was a cool, certainly a very cool and great progress by these two scientists. But you see the history and timeline. Although we might see gaps at certain positions, this is, yes, I was looking for the date, 1953. Although this started from 1869 with the interest of Friedrich Miescher, and then work by Altman, Kossel, Asbury-Bill, and then Franklin Wilkins, and now Watson and Crick. There has been continuous build over the previous work. So this summarizes a very important progress in the history of exploring the chemical composition of the molecular information that we have. Now, before moving on, there's another piece of history that I guess is very important to highlight, okay? It could be the case that we may revisit this somehow later on, but I want really to bring your attention to a very important progress that happened after knowing the double helix, okay? The double helix model by Watson and Crick, by James Watson and Francis Crick. In 1958, there was a very important progress and very important experiment by Michelson, that is called Michelson. These are the name of two scientists, and Stahl, who was working that experiment. Okay. Oops, sorry. That is what we know today as the most beautiful experiment. That is the most beautiful experiment in biology. Okay. That is the experiment that we point to as the most beautiful experiment in biology. So actually, I want to share with you some details of this experiment because it helped to understand a very important concept in biology, which is related to a question that many were curious about. Now, we have one cell. We have this genetic information that by now we know this is, we know the structure, we know it's the double helix, and many of that information. But when we see that there's a parent cell here, and then there's a daughter cell here, they were able to find that, oh, they have exactly the same information in both of them. So this suggested some DNA replication. There's a good credit for Watson and Crick because they gave an insight in their paper that they wrote they wrote a statement that implies that there is some copying mechanism. But until Michelson and Stahl came, nobody knew how that copying mechanism is happening. That is leading to having two cells having exactly the same information. Okay. So an important piece of information here that in design of an experiment that they worked on is to come up with some protocol to answer that question. And there there were three assumptions in general. There was the conservative, that was the conservative, okay, conservative view of DNA replication and copying. And there's the semi-conservative, okay, semi-conservative. And then there was that dispersive theory of how copying could happen, okay? And by then, you know, they were just different thoughts and ideas. Nobody knows until Michelson and Stahl came and they said, we're going to do an experiment to know which one is the right one. But let me explain what is meant by the conservative one. Well, if we have the genetic material here, okay, and conservative is saying, well, this would be copied here, okay, as when we go to the second generation, okay, and then a new cell, okay, somehow is going to have replicate of information in this way, okay. Semi-conservative is saying that, well, this is going to be unwinded, and so it will become unfolded into two strands, okay, and based on that, what's going to happen is I can do it this way, and okay, and this way. And I'm going to change the color here to give you an illustration. And then a complementary process is going to come here and actually build the other strand. Okay, so it's going to look something like this. Okay, that is the semi-conservative overview, that the copying mechanism is actually involving breaking down the double helix structure and doing the copying. Dispersive came up with the similar insight that this is to be cut into a certain way, okay? And the way that this would be cut, something of that sort, okay? And let me do it this way. And then change the color to give you an overview for that. Oops. So here you go, okay? So now we have three theories of how this can go. The most beautiful experiment was about designing a way to find out the actual theory through an experiment. Now, it could be that some of you attending this course, they know already that this is the actual answer, that is semi-conservative. By then, this was not one of the most probable answers, because the double helix structure, as we will study it in the coming slides, is very solid and strong, and the energy of binding is very high. So just to assume, and they knew this, to assume that, oh, the copying mechanism involved breaking this down was not very reasonable by then. So an experiment was needed. Without diving into so much details and in depth, I'm going to just highlight a couple of thoughts into how Michelson and Stahl designed their experiment, but you can read about this in the other resources. Okay. So what they did, they worked with the E. coli bacteria. Okay, that is E. coli. And then they wanted to grow this as a first generation in a medium filled with nitrogen 15, okay? So you can think the bacteria is going to feed in that nitrogen, and this is known to be a heavy material. And once they have done this, okay, they would take this, they would grow this into second generation. So this is, let's say, the first, and you're gonna move the second, and here, let me change the colors, okay, put some blue. They're gonna introduce another medium with nitrogen 14, which is known to be light, okay? So if there were, you know, the new cells, the new bacteria feeding on this is going to be labeled by specific colors. And then they went to centrifuge this, so through the machines, you know, to try to isolate the material. What they ended up with in that tube is the following, okay? After doing this whole experiment and centrifuge that, let me do it here. For the first generation, what happened is they ended up with, since this was a heavy material at the bottom of the tube, this is how it was isolated. This is the result of this, okay? Now, what about the result of the next one? After going to the second generation, the result is that they ended up having the following. I need to put different colors here. So I'm going to put purple for this area. So they had that level of color right in the middle, meaning they had half light coming from here and half heavy. So this finding here pointed out that, okay, this is certainly not the conservative approach, because conservative is going to assume always you will have the heavy material and the light material, that they're going to be two separate things. But what we see here is this purple color. We don't see the red anymore. So then they knew this is either going to be dispersive or it's either going to be semi-conservative. But the conservative theory for DNA copying is now canceled as an option. So how to resolve this? They had to go for another experiment, okay? Go further with another second round, okay? For this. And then here, what they ended up with in that case, so I'm gonna draw this now as a purple tube. Again, they had this material here, but guess what? Now, what did they have? They had now a separate new material only with the light information. And that's what suggested that this is only possible with the semi-conservative, okay? Because dispersive is going to actually keep it here, okay, guys, with some mixing. However, the moment we see this categorize into some mixing and some light cells with the light, some light colored cells with the light nitrogen, this meant, okay, the new strands came from the new generation, and now this is semi-conservative. And this really established the idea and confirmed that very important finding, that the double helix structure of the DNA actually unwinds and breakdowns in that way, and then the DNA copying mechanisms starts by building complementary information for that, okay? So I think it's very interesting to look into this history, and hopefully you will read the, look into the material shared, the reading material for the first week, and think that is very helpful. Okay, so with that, we move into the DNA, okay? Deoxyribose nucleic acid, okay? So this has, the DNA, when we look into it, is a polymer made of repeated elements that we call bases. By now, you might start knowing that we have adenine, guanine, cytosine, thymine, and uracil, which is part of RNA, okay? And for short, we refer to this A, and this as G, and this C, and this T, and this U. And this is the genetic information that we're looking at extracting at the end of the day. DNA is known to be as the molecule of life. DNA is well known to be as the molecule of life because once we extract this DNA, we have a list of alphabets, right? You can think of it as a list of alphabets. This is what here? This is giving us certain type of information, okay? Some things may refer to a mutation related to one of the diseases, and some part may refer to how we look like our hair, our eye color, and so on and so forth, and other traits. So DNA is really the reference of everything. It is the reference that shows the differences between you and I. It is the reference that shows differences between all species around us. At its core, when we refer to every single atom of those, they are composed of three important parts. The base, which could be any of these nitrogen-based parts, cytosine, thymine, uracil, adenine, and guanine. So that is the base. And the backbone of that base, so the base need a backbone, and that's going to act to build this ladder or this structure of the DNA, okay, which is the deoxyribose sugar atom, okay, and then the phosphate group. And those, you can see here, there's the base, there's the phosphate group, and then the ribose or the deoxyribose, okay, where one oxygen is omitted here as compared again to RNA. We'll come to this later on. So these are the three important parts for every single base, for every single atom in this DNA. And the list of those is going to make what is called the DNA. Okay? Now, one thing I can share with you as well here in this part, if we were to write down the structure of adenine, for example, it is this nitrogen here. Okay, 2 bonds here. There's another nitrogen here. Sometimes my drawing is not the best, but anyway, I'm going to put another nitrogen here. Okay, I'm looking at my reference notes. Here it is. And then there's an NH group here, and there's another nitrogen here. Okay, so this is the base part here. This is for adenine. Okay, now I'm going to put another structure here. Okay. This structure is made of this shape, okay, and it should be that way. And of course, this is simplified. There's a nitrogen here, there's one oxygen here, and there's an NH group here, and there's oxygen here, okay? And when these meet each other, this is thymine, this links to that. Okay, and that's what's called base pairing. We know that adenine links and base pairs always with thymine and guanine with cytosine, and vice versa, thymine with adenine and guanine with cytosine. It might be an interesting exercise giving this information to try to write down what's going to be the base pairs for ACCGTA, okay? So think about it. I'm going to give you a moment here. Try to figure it out. So for each nucleotide here, for each atom, what's going to be the paired base pair at this stage? Okay, this is adenine is going to link to what atom? Okay. So if you think about it, A would link to T, C to, sorry, to G, G, C, A, T. Okay, T, G, G, cat. Okay. So this is how we have the base pairing in this case, and it's very strong, as we explained, connection and link, especially giving these deoxyribose sugar, which gives the needed energy level, and makes the backbone very stable. So the phosphate groups, the phosphate group in this structure that you see here. Oops, let me change the color here. Sorry. Okay. Oops. So the phosphate group that we see here in this slide is imparts a negative charge to the DNA molecule, which is important to the interaction of DNA with various proteins. And it affects the solubility in the cellular environment. Sugar and phosphate backbone. Oops. Okay. Sorry, guys. So sugar. So sugar part. Okay. Sugar part and phosphate group. They provide the structure. Okay. And they are very stable. Makes the backbone very stable, and it provides stability that gives the structure for the DNA, and helps in connecting between the nucleotides. So this is very interesting way of having the chemical composition of the different bases. So highlighting DNA and RNA. DNA is the deoxyribonucleic acid, and there's RNA, the ribonucleic acid. We will come to this when we discuss as well transcription process as part of the central dogma. For the time being, what you need to know that RNA is single-stranded, usually single-stranded, okay? Usually single-stranded. However, the is double-stranded, has a double-stranded structure. That is one thing. And if you look at the sugar part here, it is deoxyribose, and for the RNA, it's ribonucleic acid. You see this oxygen here is missing here, okay? That is the difference. That's why we call this deoxyribose, which makes actually the DNA from a chemical point of view from a chemistry point of view, more stable than the RNA. And this is something very well known, okay? And eventually we have guanine, cytosine, adenine, and both of them. However, thymine is only part of DNA, while uracil is going to be part of the RNA. So in RNA strands, we don't find thymine. These are the three major differences between DNA and an RNA. Okay. So I'll be concluding for this part, and then we'll continue with the next part for the first lecture of week 1.